Hello everyone, welcome to today's lecture. My name is Dr. Aidan Seeley and I am the Deputy Programme Director for the BSc Medical Pharmacology programme here at Swansea University Medical School. But I'm talking about something a bit different today. Traditionally, I tend to go on and on about drugs. I love drugs. But today I'm going to be talking about death receptor signaling and trafficking, which actually made up a part of my PhD project a couple of years ago. So we're going to be looking at arguably the coolest name family of receptors in our genome. So today, as I say, we're going to be looking at the function and signaling of the death receptor family and looking at the mechanisms of death receptor endocytosis and the potential to exploit them clinically. So by the end of today's lecture, you should be able to compare and contrast the different mechanisms and signaling cascades which result from death receptor activation and how these receptors are then internalized through routes such as clathrin-mediated endocytosis. Additionally, you should be able to evaluate the potential for utilizing these family of receptors in a clinical setting and highlight some of the drugs that we have tried to get into the clinic which target these family of receptors. Now, hopefully you will have seen at some point in your education here at Swansea the Hallmarks of Cancer diagram by Hanahan and Weinberg. Now, this is a seminal review paper which really pulled together the vast and complex nature of what changes occur in cancer cells to make them pathogenic, to make them cancers in the first place. And there's a number of key hallmarks such as the activation of invasion and metastasis, the induction of angiogenesis, so the formation of new blood vessels in order to supply the growing tumour with all the necessary nutrients and oxygen that it needs to continue to grow. But there's also things such as sustaining proliferative signaling, so things such as EGFR signaling can become derailed, HER2 signaling can become derailed, and as a result this sends more keep growing, keep dividing signals to the cancer cells. We can also have the evasion of growth suppressors and a collection of other different hallmarks of cancer. However, for today, we are going to be focusing on the role of resisting cell death. Now, normally cells are programmed to live and then to die, and then we have the normal maintenance of tissues, homeostatic balance, ensuring that old or damaged tissues are replenished and replaced to prevent disease. This isn't the case in oncology, where cells resist cell death for too long, and so we can end up with pathogenic conditions here. And we're going to be looking at some of these underlying mechanisms and the receptors involved in those, namely the death receptors. In humans, there are 8 members of the death receptor family, arguably the coolest name for any protein family in our cells. And they've been discovered at various points in time, which means the nomenclature surrounding this family does get a little bit complicated. You can see that we have death receptors 3, 4, 5, and 6, and then some other family members which don't follow that form of nomenclature, things such as tumor necrosis factor receptor 1. However, all of these receptors do have one thing in common, in the fact that they all contain a C-terminal death domain, or DD. And this is a 60 to 80 amino acid long portion of the receptor, which allows it to interact with a number of downstream signaling effectors, driving cell apoptosis. For the purpose of today's lecture, we're going to be primarily focusing on death receptors 4 and 5, so DR4 and DR5. And this is because these are by far the most extensively characterized of the death receptor family, and is also the focus of my PhD research. These receptors are activated by tumor necrosis factor-related apoptosis-inducing ligand, or TRAIL. Now, it's no surprise that people do not tend to say tumor necrosis factor-related apoptosis-inducing ligand, because it takes too long and you'll be out of breath before you finish your sentence. As a result, we tend to refer to it as TRAIL. And TRAIL is expressed on the membrane as a transmembrane protein by immune effector cells. TRAIL can be cleaved and release a 24 kilodalton extracellular, which ultimately can then activate either DR4 or DR5 and trigger a number of downstream signaling pathways in order to activate cellular death, or apoptosis. The soluble 24 kilodalton form of the ligand is considerably less active than the membrane-bound TRAIL. So TRAIL, when it is still a transmembrane protein, is much more active than once it's cleaved and forms a soluble portion which can then activate receptors independent of its transmembrane protein. The picture is not as clear as we would like, however, due to the occurrence of what we call decoy receptors, or DCRs 1 and 2. And TRAIL can bind to these decoy receptors, but they lack the cytoplasmic death domain, which is required for apoptosis induction. Essentially, they act as a dead end. So TRAIL can bind to the receptor, but there is no intracellular death domain to allow the activation of downstream signaling cascades, and so we do not have activation of apoptosis if TRAIL activates decoy receptors, hint their name, decoy receptors. However, TRAIL will activate cellular apoptosis if it is activating death receptor 4, DR4, or death receptor 5, DR5. As a result, overexpression of decoy receptors 1 or 2 can be used as a mechanism to protect cells from undergoing apoptosis, as it acts to sequester TRAIL away from DR4 and DR5. If, for example, cancer cells then express lots of decoy receptors on their cell membrane, then there is going to be binding of TRAIL to those receptors rather than binding to the DR4 or DR5, so we're not having activation of apoptosis. As a result, decoy receptors are actually very highly expressed on healthy tissues because we have homeostatic balance. We want to ensure that our immune effector cells are not triggering healthy tissue to die. And as a result, we have a high expression of decoy receptors in healthy tissues to make sure that we do not have inappropriate cell death. Activation of DR4 or DR5 by tumour necrosis factor-related apoptosis-inducing ligand, or TRAIL, results in the recruitment of adapter proteins, including the FAD protein, or the FAS-associated death domain. And this occurs by homeotypical interactions between the death domain of FAD and the death domain of DR4 or DR5. And this is the first step in activating signal pathways and signaling cascades in order to induce apoptosis through DR4 and DR5. Recruitment of FAD will then recruit proteins known as procaspase 8 and procaspase 10. Now, the complicated thing with procaspases and caspases is we have discovered them at different points and we've not necessarily understood the pathways that they're involved in, so they're numbered slightly out of sequence just to keep us on our toes and keep us guessing. So in this case, the first things which are recruited following FAD homotypical interactions with the death domain of DR4 or DR5 is then the recruitment of procaspase 8 and 10. And these are protease enzymes, essentially meaning that they act as molecular scissors to then go and cut up lots of the proteins in the cells and start inducing the induction of apoptosis. This interaction between death receptor 4 or death receptor 5, along with the FAS-associated death domain and FAD and procaspases, forms a multi-protein complex which we refer to as the death-inducing signaling complex, or the DISC. At this point, inactive procaspases undergo autoactivation and cleavage to become active caspases. As I say, these caspases can then act as a pair of molecular scissors, and then they will start breaking down and cutting up proteins in the cells in order to then induce apoptosis. So procaspases are inactive and they are then activated to caspases at the DISC, the death-inducing signaling complex. Importantly, we have to have very tight regulation of TRAIL-mediated apoptosis in order to prevent uncontrolled excessive cell death. That can be very damaging and we can lose healthy tissues, which is not only energy costly, but can ultimately result in morbidity or mortality of the organism in question. As a result, we have evolved a number of key inhibitory apoptosis proteins to prevent the cell undergoing apoptosis. And cancer cells can overexpress some of these in order to try and inhibit cell death and ensure that the cancer cells live for longer. Examples of these anti-apoptosis proteins include things such as Flice inhibitory protein, which we refer to as FLIP. And this is a potent blocker of trail-mediated apoptosis. FLIP itself interferes with caspase-8 activation. It prevents pro-caspase-8 being converted to caspase-8, and as a result, you do not get these FAD caspase-8 dimers appearing at the death-inducing signaling complex. And without this pro-caspase activation to caspase, we do not get the induction of these molecular scissors to then cut up proteins and promote the induction of apoptosis. As a result, FLIP interaction with the disc then means that we do not have pro-caspase to caspase activation and apoptosis is halted. Downstream signaling from death receptors 4 and 5 is actually extremely interconnected and vastly complex. We have a number of pro- and anti-apoptosis proteins to either help promote or inhibit apoptosis in order to maintain normal homeostatic balance. Now that makes it extremely difficult for us to tease out the exact mechanisms underlying death receptor signaling, but we do have some clear ideas of what happens next. Once the disc has converted pro-caspase 8 and 10 into the active caspases 8 and 10, we then have a caspase cascade. So essentially, once we activate 8 and 10, they begin to activate a number of other caspase family members, such as caspase 3 and caspase 7. And an important intracellular target of these caspases is a protein called BID. And at this point, caspases then interact with BID and cleave a small portion of the BID protein to form truncated BID, or T-BID. Once caspases have converted BID into T-BID, it's then able to interact with a family of proteins known as BH3 proteins, which act to puncture the mitochondrial membrane. In this case, T-BID interacts with the proteins BAX and BAC. This interaction, as I say, acts like a molecular hole punch and punctures holes in the mitochondrial membrane, which promotes mitochondrial outer membrane permeability, or MOMP. Activation of MOMP then promotes cytochrome C release from within the mitochondria to its release into the cell cytoplasm. And cytochrome C is then able to interact with a number of downstream effectors themselves and further promote cellular apoptosis. Now, this process itself can be inhibited by a number of anti-apoptotic proteins such as BCL2, and we can also control this using BH3 mimetics by using pharmacological intervention. In this video here then, we can see what happens when we actually administer trail to cells in vitro. We can see that cells then begin to undergo cellular apoptosis, they begin to form apoptotic bodies, they begin to round up, and as a result we then see the impact of activation of these downstream signaling pathways once we administer trail to activate the death receptor proteins. Now, really the purpose of this lecture is to now look at how do we regulate and control those signaling cascades. And a key way of controlling receptor signaling, as you will be aware, is through endocytosis. So removing it from the membrane so it can no longer interact with endogenous ligands, it can no longer be activated, but we also know that that may interact or result in the activation of endosomal signaling and things like that. So it's maybe a bit too simplistic to say that endocytosis simply is there to remove the receptor from the membrane to prevent signaling, because it may actually activate a whole other plethora of downstream signaling pathways. But we're going to look at it in this context today as what happens when DR4 and DR5 undergo endocytosis. Now, hopefully you're relatively familiar with the process of clathrin-mediated endocytosis, where we have recruitment of proteins such as AP2, which results in the initiation of clathrin-coated pits. And depending which studies you're looking at and which vein of thought you have, this may either be the gradual recruitment of clathrin, which results in the bending of the membrane, or there's additional school of thought where clathrin is a flat sheet which undergoes significant remodeling in order to form a clathrin-coated pit and ultimately then begin to bend the membrane. These pits, which form clathrin-coated vesicles, will obviously then break from the membrane and are internalized intracellularly. Now, clathrin-mediated endocytosis and all routes of receptor internalization are vastly complex and interconnected, and it varies receptor to receptor, tissue to tissue, and even varies depending on how much of the ligand is in the local microenvironment. I'm sure Dr. Rowe has covered the seminal work by Sarah Sigismund, which looked at the impact of high-dose EGF stimulation of EGFR versus low-dose stimulation of EGFR via EGF. Low dose would promote the internalization through clathrin-mediated endocytosis routes, while high dose essentially results in saturation of clathrin-mediated endocytosis, and so calveolar-mediated endocytosis is then required. Importantly, whatever route of receptor internalization is occurring, it relies extremely heavily on protein-protein interactions to allow progression through receptor-mediated endocytosis. And we know this is extremely important in clathrin-mediated endocytosis, and this can be through protein-protein interactions dictated by phosphorylation or ubiquitination, which you will have heard quite a lot about due to the role of Sybil in receptor internalization through things such as EGFR interactions, and then that ubiquitin moiety acting as an adapter to allow other proteins to interact. Now, the mechanism by which DR4 and DR5 are internalized is of interest, so we can try and hijack this for oncology and try and utilize this in order to then manipulate it for clinical benefit. However, the exact mechanisms underlying DR4 and DR5 endocytosis is contended, and that was why I focused on that during my PhD. So let's take a look at the information that we do know about how DR4 and DR5 undergo receptor-mediated endocytosis. Death receptor activation promotes caspase-mediated destruction of a number of clathrin-mediated endocytosis machinery components. A 2006 study by Austin et al. Demonstrated that trail treatment for up to 2 hours resulted in clathrin-mediated endocytosis protein degradation. You can see here a Western blot analysis looking at AP2 and the clathrin heavy chain. And you can see that the AP2 protein begins to undergo a cleavage event following stimulation with trail at up to 2 hours. You can see that in unstimulated cells, so at time point 0, we only have one band of AP2 protein. However, over time, that starts to undergo cleavage because trail will ultimately interact with the disc, promote formation of the death-inducing signaling complex, which converts caspases from procaspases. And once they're converted to these caspases, they will then begin to break down cellular proteins. So we can see here very clearly that the formation of AP2 cleaved products, and we can also see this with clathrin heavy chains, that we see the emergence of these other products, these other protein products, as the time period increases up to 2 hours. And this study demonstrated that death receptor activation promoted cleavage of endocytic machinery components, as I say, namely AP2 and the clathrin heavy chain. And this was one of the first studies looking at the interaction and the co-involvement of death receptor activation and its interaction with endocytic components. Now, the 2006 Austin study also utilized a really cool experiment using the transferrin receptors. And transferrin receptors are constitutively endocytosed with or without ligand binding. So even in the absence of ligand, they will be internalized intracellularly and then reinserted, recycled back to the membrane to potentially bind to transferrin. And we can measure how well a cell can do this by using labeled transferrin uptake as a measure of the endocytic capability of the cell. And this is exactly what Austin done when looking at the impact of trail on endocytic machinery. Now you can see two lines on the graph here. Trail negative is a number of white circles which all cluster at 0 because there's no trail pre-treatment here. The second line is trail positive, the dark black line with the black squares. And they're measuring the uptake of labeled transferrin. And what you can see is over time, the uptake of labeled transferrin is reduced in cells treated with trail. And this is a result of the fact that trail begins to affect the internalization of transferrin and the transferrin receptor by extension, as death receptor activation results in degradation of endocytic machinery. It starts to cleave AP2 and clathrin heavy chains, and so we lose the constitutive endocytosis of transferrin receptors, and so the cells are not able to uptake the labeled transferrin anymore. And that's why we see up to 2 hours this steady decrease in transferrin uptake, resulting from the fact that trail activation has broken down and cleaved endocytic machinery, so we no longer have this constitutive internalization of the transferrin receptors. It's a really cool experiment. So what about the other machinery involved in endocytosis, not just AP2 or clathrin? What about the other proteins, such as those involved in the scission event at the membrane? So scission events are when we then have the cleavage of the clathrin-coated pit from the membrane to form a clathrin-coated free vesicle. Now, this is mediated by the large GTPase protein called dynamin. Now, these GTPase proteins essentially act as a molecular switch throughout the cell. Just as a bit of a recap to make sure that we're all on the same page with what I mean by GTPases, they are enzymes which are normally bound by GDP, and in this stage they are in the off conformation. We can then have the swapping of GDP for GTP using proteins such as guanine nucleotide exchange factors, or GEFs, which swap out GDP for GTP. At this point, they become on. They're in their on state and able to have their cellular function. For example, in the case of dynamin, resulting in membrane scission. Now, these GTPases may have constitutive activation to begin to break down the GTP to GDP, or this can be promoted through GTPase activating proteins, or GAPs, which promote the conversion of GTP back to GDP, and this then switches the cellular GTPase back off. It puts it from the on state to the off state by the removal of this high energy third phosphate bond in the GTP and converts it back to GDP, and then the cycle can begin anew. So dynamin then utilizes GTP in order to undergo its molecular function, which is promoting the scission event of the vesicle from the cell membrane. Now, the exact mechanism by which dynamin actually does this is relatively contended. It can do this by one of 2 proposed mechanisms, and we don't know exactly which one is the actual mechanism. Proposed mechanisms include the fact that GTP hydrolysis may elongate the vesicle neck, so essentially dynamin pushes the vesicle away from the membrane, and as it keeps pushing and pushing and pushing, eventually there will be a scission event and the vesicle snaps free from the membrane. Alternatively, GTP hydrolysis may promote dynamin constriction, where it gets tighter and tighter and tighter around the membrane until essentially it severs the vesicle from the cell membrane. Now, we don't know exactly which mechanism is actually employed, and it may very well be that it's one and the other, depending on cellular circumstance or the exact type of dynamin in question. But for the purpose of today, all we need to worry about is the fact that dynamin promotes scission events from the cell membrane. And following on from the 2006 Austin study, not very much happened looking at the interaction between the death receptors 4 and 5 and the endocytic protein families. This was until 2017 when a really cool study came out looking at the impact of dynamin 1 knockouts. And by knockouts, we're talking about cells which do not have the gene, so cannot produce dynamin-1 protein. And with knockout of dynamin-1, scission of clathrin-coated vesicles from the membrane is obviously prevented because you do not have the cells being able to then either undergo this constriction of the cell membrane until you have a snapping of the membrane, or you don't have that elongation of dynamin to push the vesicle away from the cell membrane until you have that scission event. Now, this 2017 study was really, really cool. And there's some graphs shown here looking at the impact of trail activation of DR4, DR5 receptors. Now, on these lines, there are 3 different cell types. You have wild type cells, which are able to produce dynamin and undergo normal endocytosis of receptors. You then have dynamin-1 knockout shown in the blue line. And you can see in the graphs that on the far left-hand side, when we have dynamin-1 knockout, so cells which are not able to internalize receptors anymore, we have a massive decrease in cell viability. Cells die quicker in the presence of trail when dynamin is knocked out. This may be because essentially death receptors 4 and 5 are not being internalized anymore. They're stuck on the cell membrane and continuing to form the death-inducing signaling complex, recruitment of FAD and those procaspases, which then act as molecular scissors to begin cutting up all of the downstream signaling proteins and ultimately promote cellular apoptosis. So we can see that without dynamin, we get much more cell death. We can see in the middle graph that caspase-3-7 activity is increased in dynamin-1 knockout because essentially we are then having this massive activation of these molecular scissors. And then you can see in the final graph on the far right-hand side that trail uptake, essentially measuring how much of the trail gets into the cell, is reduced in the dynamin knockout because the cells are not able to uptake the dynamin. Because they've not got dynamin, they're not able to uptake trail anymore. So it's stuck at the cell membrane, it's not being internalized, and that's why that is lower. Now, if we then express a dynamin protein, which doesn't rely on the endogenous gene, so if we express, for example, dynamin-1 tagged with enhanced green fluorescence protein, we can rescue this phenotype and you see that there is a marked improvement in cell viability. Less cells die because dynamin is then able to promote the internalization of death receptors 4 and 5 into the membrane, from the membrane into the cell, and so we see this marked reduction in the effect of DR4, DR5 activation, and ultimately we see an improvement in cell viability, and we see a marked reduction in caspase-3-7 activity, but the receptors are still internalized. And this is because we've then got this EGFP tagged dynamin allowing or enabling the internalization of death receptors 4 and 5. So this is a really cool figure showing the importance of endocytic proteins on the impact of downstream signaling from death receptors 4 and 5. Now, I'm sure you could tell that from that 2017 study, I got very excited by the really cool experiments that they had done using dynamin. However, it's probably a bit clearer for you to understand if we look at it in this diagrammatic manner. So here we have DR5 activation, and we can begin to have an internalization event following its activation by endogenous ligands such as trail. And dynamin will then promote a scission event from the cell membrane to release DR4 or DR5 into the intracellular environment, where these receptors can then either be degraded or recycled back into the cell membrane. In this case, likely going down degradation pathways to terminate signaling from death receptors. Now, if we remove dynamin then, these receptors reside on the cell membrane for much longer, and they can continue to form the death-inducing signaling complex and continue to convert procaspases into caspases, and these caspases can then go and start cutting up all of the proteins within the cells. And so you end up with induction of things such as MOMP, the mitochondrial outer membrane permeability, cytochrome C release from the intracellular or the intermitochondrial folds, released into the cell cytoplasm, and ultimately full activation of cell death. So Dynamo 1 knockouts shown in the 2017 study promotes apoptosis as the death receptors are no longer able to be internalized and so signal for longer at the cell membrane and ultimately activating more caspases from the death-inducing signaling complex. So how can we then look at what is recruited to the death-inducing signaling complex in experimental conditions? We can actually utilize a really cool way of doing this known as death-inducing signaling complex or DISC immunoprecipitations, where we utilize an antibody to pull out proteins of interest to then examine by techniques such as Western blots, which you will all be familiar with. And we can do this to try and work out which proteins are recruited to the death-inducing signaling complex following activation by ligands such as TRAIL. So you'll now be very familiar with the fact that the DISC contains the death receptor 4 or death receptor 5, as well as the Fas-associated death domain and pro-caspases, which are then activated to caspases. And we can then utilize magnetic DR4, DR5 monoclonal antibodies to bind to those receptors. We can then lyse the cells, so essentially break apart the cell membrane, and ultimately we're left with a complex which contains the magnetic antibody bound to the receptor and all of the proteins which are then interacting with the receptor. So by extension, we can then pull out that receptor and look at all of the proteins involved in the death-inducing signaling complex, such as FAD, which is the Fas-associated death domain, or the pro-caspases. And we can then analyze these by common techniques such as Western blotting. By utilizing the DISC immunoprecipitation assay, we can observe recruitment of endocytic machinery to the death-inducing signaling complex. And what you can see here is a figure taken from my PhD work, which essentially pulls out the death receptor 5 following different levels or different time points of stimulation with TRAIL. So you can see that I've treated it for half an hour, 1, 3, and 5 hours of TRAIL. And I've then utilized these magnetic DR5 antibodies to pull out the death-inducing signaling complex and probed for a number of different proteins to see when they are recruited to the DISC. Now, proteins that we would expect to be there include the Fas-associated death domain, FAD, and caspase 8. So you can see these have been detected by Western blot analysis, and those levels don't really change over time. Interestingly, we see recruitment of FLIP, this FLICE inhibiting protein known as an anti-apoptotic protein, but we also see recruitment of other well-known endocytic proteins, including clathrin being recruited at half an hour and getting stronger and stronger bands, meaning more and more protein at up to 5 hours. We can also see recruitment of AP2 with levels appearing at 1 hour up to 5 hours, and also the interceptin 1, which is a cell signaling and endocytic protein, and namely my protein of interest at the time, ITCH, which is an E3 ligase similar to Sybil. And so you can see the recruitment of these endocytic machinery components at the death-inducing signaling complex, things such as interceptin 1, clathrin, and AP2. And this occurs in a time-dependent manner following activation of DR5, because you can see that over time, those bands become more intense as more protein is recruited to the DISC. Now, you're maybe asking, what is the function of this ITCH protein? Why does that matter for death-inducing signaling complexes? Why are we talking about ITCH? Now, as I say, ITCH is an E3 ubiquitin ligase similar to Sybil, and my work has shown that ITCH is recruited to the clathrin-coated pits. You can see here some staining which has looked at GFP-tagged ITCH, forming these little puncta on the cell membrane, and these significantly overlap with the puncta present in clathrin-coated pits, shown in red. And you can see that when we overlay these images, you end up with these lovely yellow dots where there is co-localization of clathrin-coated pits and ITCH-coated pits. Now, itch E3 ligase was first identified following genetic studies of mouse coat colour, which identified a progressive and systemic autoimmune disorder characterised by excessive skin itching, hence the name itch. Since then, however, the itch protein has been shown to have a number of distinct functions, such as immune regulation, inflammation, and spermatogenesis, and has also been shown to have a role in disease states, including things like cancer cell proliferation, and also having a role in interacting with key endocytic machinery, including proteins such as Sybil, which is responsible for the ubiquitination of EGFR, and intersectin-1, which is at the intersection between endocytosis and cell signaling. Moreover, itch knockdown cells, shown in blue here again, were shown to have increased cell viability compared to those that had itch expression. So essentially, when there was less DR5 on the cell membrane, when we activated with trail, we saw less death. Makes sense? Less receptors on the membrane, less activation, less signaling, less death. And so itch knockdown cells were shown to be more resistant to trail treatment than those of their wild-type counterpart. It seems that itch knockdown cells don't seem to be able to trigger apoptosis as well as wild-type cells. So, what effect does itch knockdown have on other proteins involved in DR5 signaling, such as BID? Remember, BID is then converted to truncated BID to interact with BAX-BAC, acting as a molecular hole punch in the mitochondrial membrane to trigger mitochondrial outer membrane permeability. So itch knockdown seems to have an impact on the ability of cells to promote BID cleavage into T-BID, and this makes sense as a downstream signaling effector of DR5. We have demonstrated that there was less DR5 on itch knockdown cells and that less cells died in itch knockdown cells because essentially we weren't getting that receptor activation. So we then looked at the impact of the protein BID. Now you'll recall that BID is converted into T-BID following cleavage by caspases to then interact with BAX and BAC, which act as these molecular hole punches in the mitochondrial membrane to trigger mitochondrial outer membrane permeability, or MOMP, and the release of cytochrome C from the mitochondria to the cell cytoplasm. In this Western blot analysis then, in the second from the bottom lane, you can see itch has clearly been knocked down. We do not have itch protein in those lanes. We do have DR5, Fas-associated death domain, flip, and we do see caspase 3 cleavage. So cleavage into from procaspases to caspases. Importantly though, in the itch knockdown cells, we don't see very much conversion of BID into T-BID. So you can see that there's two bands there, the top band being the full length BID, and then the lower band being truncated BID or T-BID. And you can see that in cells that express itch, where they have higher levels of DR5 on the cell membrane, they have normal conversion of BID into TBID to induce cell death. In cells without itch, there is less DR5 on the membrane, and so we have less activation of those signaling pathways and less conversion of BID into truncated BID, so we don't see these smaller fragments appearing by Western blot analysis. Moreover, if we look at caspase 3-7 activity, we can see a marked reduction in itch knockdown cells in response to trail stimulation. Essentially, these cells are no longer able to activate downstream caspases, such as caspase 3 and 7, because we have reduced activation of DR5 at the cell membrane. As a result, if we have no TBID, you have no mitochondrial outer membrane permeability and a reduction to no apoptosis of those cells. So the role of itch E3 ligase in death receptor signaling and trafficking is an area of ongoing research. But itch has also been shown to regulate Sybil, which ubiquitinates EGFR and controls its internalization and signaling, and you will have already heard quite a lot about Sybil. What is interesting though is that Sybil also seems to have an impact on regulating the expression of death receptors 4. So hopefully now what you can see is that by understanding complex mechanisms surrounding death receptors 4 and 5, that we can begin to identify novel targets as treatments of things such as oncology, which are traditionally known to be difficult to treat due to resisting cell death. So if we can understand the underlying mechanisms of those proteins, we can then target them with therapeutics. And that's exactly what we've been doing with some novel monoclonal antibody therapies. So now that we have started to dissect the underlying mechanisms and signal transduction pathways for these death receptors, we're able to start looking at them for clinical aims. And can we essentially target these as therapeutic targets to treat diseases such as cancer? So we can stimulate these receptors for clinical benefit, as normal tissues have this high expression of these decoy receptors. And you'll recall, as I mentioned earlier, these decoy receptors lack the C-terminal death domain, so are not able to interact and form the death-inducing signaling complex. And they have a high decoy receptor expression in normal tissues compared to those of cancerous cells. So direct stimulation of death receptors 4 and 5 with agonists such as trail have been met with limited clinical success so far, because essentially we do still have some off-target effects and activation of tissue death in places that we don't want it to occur, i.e. non-cancerous tissues. We have developed a number of agonistic monoclonal antibody therapies for DR4 and DR5, such as mapatumumab, which is activating the DR4 receptor to form the disc and induce apoptosis in cells. Lexatuximab is an agonistic DR5 monoclonal antibody therapy, also activating the formation of the disc and ultimately apoptosis. Now you can tell when drugs are monoclonal antibody therapies if they end in MAB, because that stands for monoclonal antibody therapy. Unfortunately, these monoclonal antibody therapies have not yet translated to use in the clinic. Lexatuximab, the agonistic DR5 monoclonal therapy, was discontinued in 2015, and lepatumumab, the agonistic DR4 antibody therapy, is currently in phase 2 trials, but little to no clinical benefit has been seen in studies with the addition of this therapy compared to standard of care. So it doesn't look like it's doing too well in clinical trials so far, but that's not to say that these drugs will not ultimately translate to either development of other therapies or the use of these in other distinct cancer subtypes or other combinational therapies. So understanding the underlying mechanisms of these receptors is still extremely important for us to identify therapeutic targets and develop new drugs for use in the treatment of diseases such as cancer. And with that, that brings me to the end of today's lecture on the death receptors and their signaling and trafficking in cells. So today we've looked at the biological function and signaling of the death receptor family, still arguably the coolest named family of proteins. And And we've also looked at the experimental evidence and published literature surrounding the mechanisms of death receptor endocytosis and their potential for exploitation clinically. And you should now be able to compare and contrast different signaling mechanisms of these receptors and the evidence which supports their role of endocytic machinery and how these are ultimately regulated following activation of these receptors. And you should be able to evaluate the potential use of exploiting these death receptors for clinical benefit. Put up some key studies and a review article on the death receptors for you to review if you want some additional information on this lecture, as well as to help consolidate any of the information covered. Of course, if you do have any questions about this lecture, please do get in touch with me. My email address is shown on screen. I hope you've enjoyed this lecture. Get in touch with any questions you have and stay safe. Bye for now.